Okay, hello everyone and welcome. Happy National Science Week. Before we start, I would like to state that WIN Australia acknowledges the traditional custodians of country throughout Australia and their connections to land, sea and community. We pay our respect to the elders, past and present, and extended respect to all Aboriginal and Torres Strait Islanders people today. Welcome everyone to today's webinar on how nuclear can save your life and the environment. Women in Nuclear Australia Incorporated, or WIN Australia, is extremely excited to be sharing this webinar with you today. In case you are not familiar with WIN Australia, we are a not-for-profit association of women and individuals of other genders who work professionally in various fields of nuclear technology and radiological applications in Australia. One of the aims of WIN Australia is to promote understanding and public awareness of the benefits of peaceful nuclear and radiological applications amongst women and young people. WIN is an international organization with members from over 100 countries. Today's webinar is supported by Inspiring Australia as part of National Science Week. WIN Australia would like to thank our sponsor, the Australian Nuclear Science and Technology Organisation, ANSTO, for its ongoing support of WIN Australia and its objectives. WIN would also like to acknowledge the ongoing support of Australian Institute of Nuclear Science and Engineering, ANSTO. We would also like to let everyone know that this webinar is being recorded and will be distributed to those who are unable to attend today and may be used by WIN for promotional purposes. No one in the audience is visible to us today. You can see us, but we can't see you. So you do not need to be concerned about being recorded. This is last in the series of webinars for National Science Week being hosted by WIN Australia. My name is Patricia Gadd and I'm a WIN executive member. I also work at ANSTO as an instrument scientist. The purpose of today's webinar is to discover how nuclear science and technology makes a critical contribution to the well-being of the planet and the people who live on it. Please use the Q&A function at the bottom of your screen to ask us questions throughout the webinar. Please remember to be kind and respectful to us and our guests. WIN has managed to find some experts today for this webinar who spend a lot of time carrying out work using radioisotopes to benefit people and the environment. I would like to welcome Dr. Elizabeth Bailey from Royal North Shore Hospital and Dr. Cath Hughes from ANSTO as our speakers today, as well as Mr. Mark Moore and Dr. Tom Cresswell from ANSTO, who will be joining us on the Q&A panel. First to present today is Dr. Elizabeth Bailey, who is the Chief Nuclear Medicine Scientist at Royal North Shore Hospital. She has more than 25 years of clinical and research experience. Recently, she has been using radioisotopes for diagnostic, therapeutic, and imaging purposes for various tumors and cancers. Over to you, Dr. Bailey. Perhaps we'll let Dr. Bailey sort out her technical issues. Would Dr. Cath Hughes like to go first?
Sure.
Okay, so
I can do that.
Dr. Cath Hughes is the Senior Researcher and Program Leader at ANSTO. Dr. Hughes uses stable isotopes and artificial or naturally occurring radioactive isotopes as environmental traces to trace water, sediment, and contaminant movement in various water bodies. Thanks, Dr. Hughes.
Hi. I thought I was going second, so I'm going to refer to a few things that assume that Liz has gone first, but that's going to be fine. I've been at ANSTO. I had a bit of an introduction from Patricia there, but I've been at ANSTO for 20 years. One of the things I was going to say in association with this photo is that I've done field work in some of the most uncomfortable places, from smoky engine room of a fishing boat in Thailand to standing for hours on a snowy creek bank in the middle of winter, waiting for water to flow very, very slowly. Today, I'm going to present to you today one of the many nuclear-based techniques that can help save the environment. Maybe. There we go. So, nuclear, good for the environment? I know what you're thinking. When people think of nuclear in the environment, the first thing that comes to mind is the atomic bomb, radioactive waste, blinky. But just like radioisotopes are used to find disease in the human body, which you'll probably find out more about in a minute after Liz goes second, they can also be used to track pollution in our environment. Metals and sediments from mining, sewage and industrial outfalls, chemicals from industry and agriculture, urban pollution and plastics in our oceans, these are really big problems in our environment. Pollution in our natural environment stresses the plants and animals and microbes that live there, and the pollution can be transferred to humans through the food we eat. We can use radioactive traces to understand how pollution behaves in the environment, from the tiniest animal or plants in the lab to big open lakes or coasts. This can help us to conserve biodiversity, improve water quality, which is responsible for providing us with clean air, clean water, and food. So what is radiotracing? Radiotracing is a technique where we introduce a very small amount of a radioactive isotope or radioisotope to trace the movement of something, almost anything in the human body, in industrial systems, and in our natural environment. Elizabeth's going to talk about medical applications in humans, so I'm not going to go any further with that. But radiotracers can be used in industry and lab environments to study mixing, transit time, flow pathways, contaminant absorption. In some cases, like this one you've just seen on the screen, the inputs and outputs can be measured of a system. In others, imaging of traces can show how things flow or disperse or spread out. Similar to human health and industrial applications, radiotracers can also be used to study contaminant uptake into plants and animals in the lab. In this example from Tom, prawns or shrimp exposed to radiotracer cadmium-109 were shown to concentrate this toxic metal in the hepatopancreas, which is like a liver, but located in the head. So don't eat prawn heads if you're not too sure where they've come from. So what are radioisotopes? Some of you will already know, and some might not be quite so sure. They are radioactive forms of an element which sometimes occur naturally, but can also be made in a nuclear reactor, such as our opal reactor, which gives them extra neutrons in their nucleus. In the case of carbon, I've just used this as an example, carbon-12 is the most common. Carbon-13 is a non-radioactive isotope that has an extra neutron in its nucleus, and carbon-14 is a radioactive isotope which is made both naturally in the atmosphere by cosmic rays, but also in a nuclear reactor or in nuclear weapons testing. So this version of carbon-14, is often used for radiocarbon dating of old artifacts. Some of you might already know about that. Chemically and biologically, the radioactive element behaves the same as the non-radioactive element, but it gives off a radioactive signal. Most radioisotopes we use as radiotracers have a short half-life of days or months, which means they decay or disappear very quickly. The huge advantage to using radiotracers in pollution studies is that they are unique with no natural background, and they can be used as a direct analog for the chemical or contaminant under study because they behave the same. Because they're so easily detected, we can use a really small amount, so there are no toxicity or concentration effects. If you use the non-radioactive isotope of the contaminant or the chemical that you're studying, you'd have to use a huge amount to be able to measure it. And if you were studying living organisms, the amount could have a toxic effect and cause it some injury or potentially make it die. And in physical systems, the difference in concentration from the area with the tracer and the area without it can alter the process that you're studying. Another advantage, I guess we've already mentioned though, is the ability to easily measure or image the tracer in situ or non-destructively, so that you don't necessarily need to take samples in order to measure it. You might be able to image it in very effective ways. And because the tracers have a short half-life, you can conduct repeat experiments without worrying about residual tracer. So listed here are some examples of isotopes used in environmental studies in the lab and in coastal and aquatic environments, in hydrodynamics, contaminant dispersion, in sediment transport, biological and chemical uptake. And you can see a whole list of different elements there that can be relevant. Many of them are quite toxic things, and some of them are nutrients that we're really interested in how they behave in our environment. And some of them are medical isotopes. So traditionally, to measure the uptake of a contaminant over time into aquatic animals like fish or prawns in the lab, you'd have to use hundreds of animals, which you dissect to measure the concentration in each organ of the animal. It's really important to say here that that all of our aquatic animal studies are conducted with animal care and ethics approval, and that we treat all animals humanely. The radiotracer approach has a great advantage of using way fewer animals, as I'll show in the next slide. So here I'm going to show you the method that Tom Creswell uses to study contaminant uptake into aquatic animals. So here metal radiotracers are added to the water, in this case with a tadpole. But it can also be done with crabs or snails or fish or other small organisms. The tadpole uptakes the metal primarily through its gills while it's breathing, and it's in a basket that can be removed without disturbing the tadpole. The live tadpole is removed from the exposure chamber and rinsed in clean water to remove any radiotracer from the outside. And then it's transferred to a small bottle very carefully with a small amount of water so it can breathe. And the bottle is placed on top of a gamma spectrometer. The instrument detects the gamma radiation coming from the tadpole and provides us with a peak, which can be converted to how much metal each tadpole has in it at that particular time. The really great thing about this method is that the tadpole can be popped back into a fresh radioactive solution, and we can repeat the process the next day. This enables us to work out how quickly the tadpole accumulates the metal over time. And the other great thing about this method is you can use multiple radiotracers at the same time to see how real-world mixtures of pollutants affect aquatic animals. Some of the animals can be preserved frozen after euthanasia and sectioned for imaging using autoradiography. Again, you'll see images like this in Liz's talk later. In this really cool example from Tom and his colleagues, selenium from coal mine waste was traced in tadpoles as they metamorphosed into frogs, which was really, really amazing. So you can see here in these pictures, first cute little tadpoles and then slowly losing their tail, growing legs and turning into a frog. And you can see where that contaminant moves through that process and where it stays in the body. So using radiotracers, we can use fewer animals and get much better data about the time it takes for a contaminant to accumulate and where it goes in the organism. This allows us to modify environmental protection guidelines to better protect aquatic organisms. So far, I've been talking about small-scale applications in controlled environments in the lab, but radiotracers can also be used to study similar processes outside the laboratory or industrial plant. Radiotracers can be used in traditional engineering and ecological applications for coastal and aquatic management. We can study the movement of sand near beaches during storm events, as well as the movement of dredge spoil or sediment. Radiotracers are the best possible tracer in these environments because they can be detected easily in the field, even when they've been buried. We can study the movement of pollution, for example, sewage or industrial discharges into coasts and lakes. Sodium iodide detectors can be towed through the water column to build a 3D picture of the tracer movement. Radiotracers are especially effective in highly polluted systems where fluorescent tracers have a high background from waste, or for tracing specific components like grease or particulates. We can study the movement of contaminants in groundwater, adjacent to creeks or discharging into the ocean. For instance, where treated effluent is injected into groundwater. And in this example, the movement of the tracer over 4 days was measured using detectors that could show what direction it was moving in. Knowledge of the fate and impact of contaminants in the natural environment has traditionally come from studying highly impacted environments that are or very polluted, or from radiotracer studies in the lab, like the ones that Tom does and that we've just talked about. With radiotracers, we can study the partitioning of contaminants such as metals, nutrients, or nanoparticles by releasing minute and ecologically harmless quantities of their radioactive isotopes into natural systems. We can see how the tracer moves through the water, whether it's absorbed to sediments, which plants and aquatic organisms take up the tracer, and how long that all takes. You might be asking, but how is it safe to put radioisotope traces in the environment? One of the things that's also important to know is that there's radioactivity in the environment already. There's quite a lot of it naturally occurring. And radiotracer studies are always designed to minimize the radioactivity used in the exposure to both the workers conducting the study and to members of the public who may be exposed. We also model the radiation dose to non-human biota independently, and recent work has been done to evaluate doses for past studies and compare them to guideline values. Generally, we've found that most radiotracer studies can be done with no adverse environmental impact, and biota dose assessments can be used to optimize the use of radiotracers. It helps that we would normally use just a few grams or even milligrams or micrograms of tracer because it's so easy to measure, and that these tracers have a really short half-life in most cases, which means they decay away to nothing quite quickly. Here's an example from Hong Kong where tritium and gold-198 were used to study the fate of treated sewage effluent discharged into a typhoon shelter in Hong Kong Harbour. Tritium, which is the radioactive isotope of hydrogen in the water molecule, was used as a conservative tracer of the water component, which means that it follows the water exactly. And then gold-198, which has a short half-life of 2.7 days, was absorbed to particulates in the sewage. And then the traces were injected in the sewage treatment plant over 24 hours and detected using sodium iodide detectors, running profiles through the water column, and then sampling was conducted for tritium. Tracer surveys were conducted at every low and high tide. And you can see here, I'm not sure, presume you can see my mouse wandering around. The tracer showed the horizontal movement, so this is a plan view over time, and the dispersion or the spreading out of the tracer as the period of time passed. You can also see this was discharged, fresh water discharged into a salty environment, and this fresh water floats across the top, and then you can see the gradual vertical mixing and stratification of that tracer. The ratio of the tritium to the gold can also be used, or was also used, to indicate the settling of particles. There are many new areas both in technological development for radiotracing and also contaminant pathways and issues to be investigated in our coastal and aquatic environments. The fate of microplastics in the environment and their impact on the food chain is one important area of research. Nanoparticle tracers have also been developed for use as water and sediment tracers. The fate of nanoparticles from industry and from cosmetic products discharged into our waterways can also be investigated using nanoradiotracers, such as cerium-141, to understand their physical transport and how they behave in aquatic ecosystems. In this example from Tom Creswell, nanocerium is used as a diesel fuel additive. He and his collaborators at CSIRO wanted to answer the question of whether it crosses cell membranes and becomes a bioavailable in different forms. So using radiotracers made in a nuclear reactor, we can make a real difference in understanding the fate and impact of pollution in our environment. These aren't the only examples of the kinds of applications we can do. There are many other kinds of applications in managing water resources, studying the age of groundwater, looking at past climate and understanding climate change, and much, much more that I'm sure you could find a lot of interesting things on our website if you want to explore. So that's the end for me.
Thank you, Cath.
And I'm going to stop sharing.
That was a very interesting and informative talk, and so lovely to hear about the many uses of different isotopes and how useful they can be for our environment. So I guess now we will try Dr. Elizabeth Bailey to see if our technical issues have been solved. Can we hear you now?
Can you hear me now?
Yes, we do. Fantastic. So now I present Dr. Bailey, who is the Chief Nuclear Medicine Scientist at Royal North Shore Hospital.
My apologies for the sound issues before. Public health Wi-Fi has its moments, unfortunately. Thank you for the kind invitation to present today. So I'm going to take a different slant to Cath's very interesting conversation. I'm going to talk about how we use it in humans and how we use it in the clinical environment. So nuclear medicine is a really important imaging modality and therapy modality that's used in medicine every single day of the week. So we use radioisotopes, radiotracers that we get from ANSTO, some of our collaborators overseas and other facilities within Australia. We make and label some of those products within our own laboratories, within our own departments, and we make products that we can either inject into a patient, they can inhale in the form of a gas, or they can swallow it in the form of a capsule. So for something like treatment of thyroid cancer, we give them a capsule that contains radioactive iodine that will actually target their thyroid cancer and kill off their cells. Then we can use specialized imaging equipment. And it can be either in what we call 2D or 3D mode. So what I would say the difference is between the two is 3D is like a 3D printer. Okay, so you get it in 3 different planes and you can see the whole of the organ in 3 different planes. And then what we're basically looking for is different patterns. And those different patterns represent different functions of different organs within the body that are specific for the question that's being asked for. And nuclear medicine has the potential to detect diseases prior to any changes that you may see on a standard X-ray or a CT scan. So I'll try and explain to you exactly what that means. So I'm sure everyone's seen a CT scan, so a CAT scan of the chest or the tummy, where you see lovely detail of the vessels and the bones and the airways of the lungs, and lots of detail in that picture, as you can see, you would see on an X-ray. The difference between that and nuclear medicine is ours is a little bit more like that. So ours is a little bit blurry. Often we're injecting something that goes to only one particular organ in the body, and that can be a really small organ. For example, an adrenal gland, which is about 10 to 20 millimeters, that sits just above the kidney in the body. So really small things that we're trying to see. When we inject the tracer, we get what we call a hotspot. Well, we can see a particular item within the body, but we really don't know where that is. So what we do is that we actually take what we do and we overlay it with that beautiful anatomical data that we get for things like CT and MRI scans. And then we can look at the function of that organ. We can tell somebody exactly where that is. And so if they need to have surgery, the surgeon can then go and remove that particular organ that may not be functioning as well as it should be. It could be a cancer, or it could be telling someone exactly what bone in their hand or their foot, for example, may be broken that may need to be repaired. Perfect example of 2D imaging that I was referring to before is a simple example here of a person who came in for they had a sore hand. They went and had an X-ray of their hand. They then came to the nuclear medicine department. We gave them an injection of a tracer called HDP, which is a bone targeting agent. So it's got bone phosphates in it. And so when we inject it into the body, it's actually taken up by the cells in the body that are actively trying to repair any damage within the body. And because this person actually had a fracture at the base of their thumb, we were able to see that as a hotspot on our scan. What we're able to do is then overlay that with the anatomical image, and we can say that that hotspot is actually in the scaphoid bone. And for the referrers that are actually trying to treat this patient, that's a really vital piece of information, because depending on what particular bone it is determines what type of treatment the patient should get. And we can become even more complicated. So this is an example of a patient that came to us with, we were looking for a particular mass in the chest. So we did a CT scan. We then got this scan. We injected a tracer called Sestamib. It's used for 2 different types of procedures. It's used to look at the function of the heart muscle. And it's also used to look for something called parathyroid glands. Parathyroid glands, you have anywhere from 4 to 8 of these. They're very tiny little glands that sit in the neck. But at birth, sometimes some of these glands, when you're actually in utero, these glands actually sit in your chest. And when you're born, they migrate up to sit in the neck. But sometimes they don't migrate up. And so we get these tiny, we see this little hotspot here, but we don't really know exactly where that is, so the surgeon can go and remove it. We pop it on with that CT scan and we can say exactly where that lesion is. So when the surgeon goes in to remove this thing, they know exactly where they're targeting. Now, as I was saying earlier, sometimes we give these injections and we get all these blobs, as we call it, on a screen. We don't really know what we're looking at. This particular scan is called a sentinel node scan. This is used for patients that have breast cancer, also in patients with melanoma. And so what we're doing is we're injecting a radioactive tracer labeled with technetium. It's something called antimony colloid. And it's tiny little colloidal particles that get trapped in the lymphatics of the body. So the lymphatics is the draining of fluid in the body. So the system, our bodies are very good at maintaining a good fluid balance. However, it can get out of whack occasionally. The other thing is that people that have cancers, like breast cancers, they can actually get spread of this cancer from where they've got the tumor in their breast into their lymphatics. As a way of maintaining fluid within the body, these things drain into lymphatics. So what this test does is we inject the radioisotope around the tumor in the breast, and we wait a few hours, and then we take a picture. And what these are lymph nodes that are draining fluid from this breast cancer that may actually have breast cancer cells in them. And for the patient, that's very important because you want to remove any cancer cells potentially that the patient may have. There are many, many lymphatics in the chest. For the surgeons that are removing the tumor, they need to know exactly which lesions or which nodes are draining from this tumor. So by injecting this tracer and taking these pictures, we're able to tell them exactly where they are. Now, of course, the other thing that people may or may not have heard about is something called PET. PET stands for positron emission tomography. It's a different type of imaging tool. It's used predominantly in the diagnosis of cancer. It detects a different type of gamma emission. So it's detecting something called positron emission. So what happens is that we inject a particular type of radiotracer. It tends to predominantly be either fluorine-18 or gallium-68. Once it interacts with a cell, it causes an annihilation. And as part of that annihilation, it emits two positrons that are opposed at 180 degrees to each other. Then we can detect those annihilations or positrons on our PET cameras. Okay, so at the back here, we have a series of detectors. These are made of lutetium oxysilicate, and they're able to detect these positrons that get annihilated once they interact with a cell, and then we can identify exactly where they are in the body. So that's what we call radionuclide therapy. So we get these particular beta tracers. We can either label them in-house or we purchase them from some of our commercial suppliers like ANSTO. We give them to the patients. It can be either injected or for something like iodine, they swallow it in a capsule, and it targets a particular cell within the body. It causes a DNA strand break and destroys the cancer cells. And there are different types of radionuclide therapies. One of them that's been around for 30 or more years, more than I've been in the profession, that's for sure, is something called radioiodine treatment, and it is very, very successful. It's routinely used in the treatment of thyroid cancer still to this day. Obviously, they make small modifications based on new research that is available, but it is still routinely used to treat thyroid cancer. And the new types of radionuclide therapies that are coming into play are labeled with these lutetium products that we get from ANSTO. One of them tags with a somatostatin receptor and the other one with the prostate cancer product. So nuclear medicine and radioisotopes, they play a really important role in the care and management of patients. We're able to have a significant impact on the outcomes for these patients. We can improve their survival so they live longer, and we certainly have a significant impact on their quality of life. And the use of radioisotopes in medicine is strong and increasing, especially in the realm of therapy. Thank you very much for your time.
Thank you so much for that great talk, Dr. Bailey. It's amazing the work you're doing in terms of using radioisotopes for diagnostic imaging and therapeutic purposes. Thank you so much. Okay, now I would like to welcome our panelists, Mr. Mark Moore and Dr. Tom Cresswell. Please join us. Mr. Mark Moore is the General Manager of Maintenance at ANSTO. He has over 28 years experience in the manufacture of radiopharmaceuticals and radiochemicals for medical imaging diagnostic use. And Dr. Cresswell is an environmental research scientist with a passion for understanding how humans impact our environment. Tom uses nuclear techniques to very precisely study how contaminants interact with aquatic animals, organisms. Okay, so we have a few questions that have been coming in. So the first one that we would like answered live is how much waste is produced in the manufacture and then in medical procedures? How long does radioactive medical waste last for and where is it stored? I think this might be for you, Dr. Bailey.
Oh, lovely. So look, it depends on the half-life of the product that you're injecting. So something like gallium-68 has a 68-minute half-life. So in fact, within a really short period, that radioactivity disappears completely. However, something like lutetium, for example, has a significantly higher half-life. The problem, of course, with some of these products is that because we have to add chemicals to them to end up with the product that we need to give to the patients, some of these products actually increase the shelf life of having to store these products. We have a waste area out the back of our laboratories that is shielded, that we store all of our medical waste. I think Mark can most probably answer how waste is handled at ANSTO, but from a patient perspective and a medical clinical perspective, we have a laboratory that stores our waste. We also have most hospitals have separate waste storage areas, usually down in the basement of the hospital, to store the waste until it decays away. Once it's decayed away, it will just get disposed into general waste.
Thank you so much. Mark, would you like to add to that about how waste is stored at ANSTO?
Yeah, from a manufacturing perspective, we do produce waste in order to manufacture nuclear medicine for Australia, but ANSTO has some very specific waste handling and management policies in place. We have waste management facilities, so all the waste that we have produced in the last 50 years still remains on site. So yeah, having a nuclear reactor, it uses fuel, it uses uranium to create the fission to produce radioisotopes. That fuel we can store. We can also send it to get reprocessed, and then we accept it back again and we store it on site at ANSTO. So there is a very long supply chain for waste management, handling, and control. And we're also a very highly regulated organisation. So we have a lot of rules to follow in the waste management of nuclear waste. And so it is stored in very specific areas under tight controls. But it doesn't produce a lot. And then there's, again, as Dr. Bailey mentioned, it depends on the isotope that we're manufacturing. Some are very short. So the main one, technetium-99m, that's used in a huge amount of diagnostics is actually got a 6-hour half-life. So after 10 half-lives, or 60 hours, you can go out in normal waste. So it's quite an easy one to handle. But then there are some of the longer-lived therapeutics that we have to keep for up to 12, 15 months before we can release those. So yeah, it's quite specific to the isotope we're handling.
Thank you, Mark. I have one for the environmental researchers. So when radio tracers are used in environmental studies, how do you know that you are measuring your radio tracer and not something naturally radioactive that was already in the environment? Tom, would you like to take that one? Or would Cath like to take that one?
That's probably a Cath.
Are you throwing to me?
Right to you there, Cath.
One of the things about the radio tracers we would use in an environmental setting the gamma emitting radio traces that are just not naturally occurring in that environment. The natural background of radioactivity is quite low for detecting that kind of radiation. So we then would choose very short-lived traces that have a distinct signal for the short period of time that we're doing the study, but following that they will have both decayed in terms of their short half-lives, but also dispersed if it's in an open water environment to levels that can no longer be detected. So yeah, so I guess that's it. I didn't actually see that red, so I can't actually reread the question to make sure I've answered it correctly.
I might just add a little bit just to say that if we are looking specifically for a certain suite of radioisotopes either naturally occurring out in the environment, we'll always take background samples and do a background subtraction so that we know what we're looking at is elevated above what that natural activity is of that specific radionuclide. If it's human-made, then obviously there's very little chance of it that it's going to be there in nature, but we'll always look for it first and try to quantify it before we say that it's elevated or not.
I can also add to that in some natural environments there are elevated levels of some radionuclides, particularly those are associated with the natural uranium and thorium series that occur in our environment, and you see those particularly in places where there's been mining. So I did a study once in Thailand of sediment transport that was outside an estuary where there was tin mining in the catchment, and there was quite elevated background because of thorium that had been deposited outside this estuary.
Thank you, Tom and Cath. Okay, I have another one. After a patient takes a radioisotope tracer, what is the process for the patient in terms of waiting for the radioisotope to decay and excrete from the body? How much radioactivity would they be emitting? And is the patient isolated? And I'll combine another question with that. What about their sewage waste?
Oh my gosh. So again, it depends on the tracer that's being emitted. So as I mentioned, gallium-68, the PET tracer has a 68-minute half-life. So in fact, a lot of these products are biologically excreted through normal body functions. They are either excreted through the urine, they're broken down by the liver and excreted out through the feces, or they just decay away. The patients, if there are particular restrictions that they need to follow. My apologies, my phone just keeps on ringing. Apologies for that. Yeah, so the patients do get given particular instructions depending on what they've actually been given. There are very strict guidelines that are released by our PANSA and the Environment Protection Authorities that tell us what levels of radiation are safe for a patient to be discharged with. If a patient is being given a radioactive therapy treatment such as iodine, which has a longer half-life, the patients do get admitted for a period of time in a specially designed therapy room that has lead shielding in the walls. And they are discharged once their rate is below the level that is deemed appropriate by the regulators. In regards to sewage waste, remember that there's a dilution effect. So if somebody, they say, for example, radioiodine, where the patient is given high levels of radiation, they're admitted for their therapy. It is excreted by biological processes out through the urine. Their urine is radioactive for a period of time. We obviously instruct the patients to double flush, triple flush the product down the waste sewage system. But by the time it reaches the outlet, there is a dilution effect. So there are literally hundreds of thousands of litres of water with a very, very, very small, insignificant amount of radiation in that dilution effect. So it's that dilution effect that when it hits the exit point, it's no longer radioactive. Does that make sense?
Thank you. There were so many questions in that one.
Can I follow that one up?
Yeah,
so we've done studies of radionuclides in sewage effluent and iodine 131 in particular. And one of the first things I'd say is that most of the major hospitals for their major iodine 131 treatment have a special tank in their basement where when they're in their special lead-lined room, that there's actually a little toilet tank as well, which holds onto that sewage effluent for a significant period of time to let it decay before it's released. But in the sewage system in Sydney, you can occasionally detect iodine 131 that's associated with that. And we've looked at the dose, the potential dose to both biota in the ocean, where the ocean outfalls are, and also to humans, and found that the radiation impact of that is negligible. Like it's absolutely unmeasurable. So if anyone's ever interested in that, I can point them towards the paper that we published on that.
And in fact, most departments no longer put those sewage holding tanks in place because they have to be managed. They've then got a very high concentration of radiation in a contained environment, and there have been documented reports of them actually leaking and shutting down entire floors of hospitals. So in fact, these days they tend not to put them in because it's actually a bigger risk than not having them in at all.
Thank you. I have one for you here, Mark. How is nuclear medicine made?
Oh, that's a big question.
So there's a number of ways we can manufacture nuclear medicine, and again, it depends upon the isotope. So you can have neutron-rich isotopes that Liz spoke about, which are formed in a reactor by putting a target into the reactor. It's bombarded with neutrons, or we can have proton-rich that Liz also spoke about, and they're made in a cyclotron. So the cyclotron speeds up protons very fast, smashes them into a target that's a different type of target. So you can have both metal targets. So typically speaking, most targets that go into a nuclear reactor are either metal or metal salts that we irradiate, and then we do a synthesis of that. We take it out of the reactor, it goes into a shielded hot cell. We then have a chemical process by which we extract what we want out, purify it, quality control it, sterilise it, goes out to the hospitals. That's a really short way of talking about it. But we can also do it via cyclotrons, where we bombard the cyclotron. Typically in cyclotrons, they can still be either metal or they can be liquid targets, such as enriched water to make fluorine 18. So it's enriched oxygen 18 water to make fluorine 18. And then you're just basically stuffing another proton into that to make it radioactive and unbalanced. From that, we then label a sugar to it, which is fluorodeoxyglucose. We then sterilise it and send it out to the hospitals where they use that to inject the patients for scans. So there's a number of different ways we can do these things. Like the number one isotope that we manufacture, being technetium-99m, is a daughter product of a whole other isotope called molybdenum-99. Molybdenum-99 is actually manufactured by irradiating enriched uranium in the reactor. We take that out, we digest it, we can then separate molybdenum out, which is 6% of the total ionizations, into a very high quality, very pure solution that goes onto a technetium generator. The generator then, through a period of decay, produces technetium-99m that we can elute off in a saline solution that can then be mixed with other chemicals like sestamide that Liz also spoke about for heart scans, for example. And we have a whole series of different compounds that can do different organs. It can do bones, heart. I think then you get the Technigas stuff that is used for lungs. So there's a whole range of different modalities that can be used for this sort of stuff. So yeah, it's quite a complex, it's a very complex question, how we make it, because we do lots of different things depending on what it is that we're making. But they all have a process of irradiation, purification, sterilisation, quality testing, and then hospital usage or whatever the therapy or diagnostic may be.
Thank you, Mark. You answered that question very well. So I have another one. Do the radionuclides specifically target the cancer cells, unlike cytotoxins, where no other body cells are affected?
They predominantly go to the cancer cells. There's always an element of the product that we inject will actually also go to normal tissues. we try and eliminate that or avoid that to the best of our ability. So, for example, the lutetium therapy that I was talking about, where we treat people with these neuroendocrine tumors with a somatostatin receptor, to avoid irradiating the kidneys extensively, we give them something called an amino acid. And the amino acid increases the rate of glomerular filtration, or the washout rate of the kidneys. So in fact, we actually clear the kidney activity out quickly. So we can give products to increase the washout from the body of activity that may be occurring in a normal organ.
Okay, it's 5 o'clock, which means we'll have to end our webinar here today. And so thank you so much to our speakers and panelists for sharing your knowledge and expertise with us and all those difficult questions. Thank you to the audience for joining us. I hope you were able to learn as much as we did. If you didn't get to have your questions answered by our panel, please send those questions through to us at winaustralia@gmail.com, and we'll send them through to the appropriate experts for an answer. So when you close this webinar, you'll be prompted to fill a survey about today. This survey is not for Women in Nuclear. It is for researchers from the Science Communication, Outreach, Participation and Education Group at the University of Sydney, who are conducting a study into the experiences of online science communication events as part of this year's National Science Week, and is endorsed by Inspiring Australia. So thank you very much, everyone, for joining us today. Hope you all have a lovely evening, and I hope you're having a great Science Week. Bye.